So the narrowest spectrum would be the best. So unison, augmentin would be better if it covers. Another thing is one agent is better than two agents. Not always necessarily. In this case, Zosyn is definitely broader compared to the combination of ceftazolime and flagyl. Okay. So we'll continue to practice, but if you get those options, like basic points. And another thing is, some noted that Zosyn or the agents you gave me would be good as empiric agents. At this point, you already have a culture and sensitivity data. This is not empiric therapy. This is definitive, or this is targeted agents you are choosing. Okay, so I didn't deduct any points, but also for rationale, you do not want to say you want to say more specific. I saw a lot of them, the positive and negative, and that's pretty much everything. You want to rationalize, Zosyn or unison would cover Klebsiella, MSSA, two bacteroides, and then Prevotella, which are negative anaerobes. So you want to provide a more specific rationale. That's how you continue to develop better decision process. And then you can also continue to narrow down better agent to optimize patients. So throughout the course, I'll bring more cases, kind of apply your knowledge to the real world scenario. Thank you for your
Okay. Well, we're going to go ahead and continue then with our discussion on targets, molecular targets in bacterial cells that our antibiotics go after. But before we do that, let's just quick recap what we did on Monday. Really quick, if this is a chromosome here in a bacterial cell, there's a couple enzymes that we can actually target with small molecules that will modulate the bacteria's ability to express their genome or replicate their genome. What was the name of one of those two enzymes that we made mention of on Monday that modulate DNA expression? Yes. Gyrase, good. What was the other one? Yeah. Good. Awesome. And we know that if we inhibit one or both of those enzymes, we can impart a lethal effect on bacteria. And what class of antimicrobials actually inhibits one? Geez, oh, Pete, come on. I tell you, I am functionally impaired when it comes to technology. Dr. Schneider would be very disappointed with me right now. And of course, from current, well, it erases everything. All right, we said gyrase and topo 4, and then what class of antimicrobial targets those? Yes, fluoroquinolones, very good. Okay, all right. There is an arrow pointing here on your blank sheet, and some of you may be asking, what is that actually pointing towards? It's pointing towards something that's not there, but I'm just going to cartoon it in here. There is an enzyme that bacteria have. It's called a DNA dependent RNA polymerase. All right, think about that name for a second. DNA dependent RNA polymerase. This enzyme is using DNA as a template to make RNA. Okay? And we have a class of antibiotics. They're called the rifamycins. The prototype is rifampin. We won't talk about rifampin in much detail until we get to tuberculosis. But that arrow is pointing to an imaginary DNA-dependent RNA polymerase enzyme, and a drug like rifampin is capable of inhibiting that enzyme. If it inhibits that enzyme, the bacteria are unable to produce RNA. That suppresses then protein synthesis, and you can actually treat an infection with rifampin. As a side note, we don't use rifampin as monotherapy very often for most bacterial infections. It is used for latent tuberculosis infections. Outside of that, have you seen rifampin used alone or in combination recently, Dr. Lewis, in your practice? Yeah, so it's got very, very specific uses. Okay. We did discuss protein synthesis very briefly. We chopped up the bacterial ribosome into 3 zip codes, if you will. We said there's an A site, there's a P site, and then there is an E site that is created when the 50S and 30S come together. We have small molecules, we have antibiotics that target specific areas of this 70S ribosome to modulate protein synthesis in bacteria. Does anybody recall the name of any of the antibiotics that bind to some place in the 30S ribosome? Talked about a couple of them. Yes. Tetracyclines, very good. So we have tetracyclines. And where do tetracyclines like to bind? What site? A site, P site, or E site? Next to P. So it's the A or the E. So the tetracyclines will actually occupy space in the A site of the 30S ribosome. And remember, by virtue of them being there, they prevent an incoming amino acyl transfer RNA from delivering its cargo, that next amino acid that shuts down or inhibits protein synthesis. Right? What's the other molecule that we made mention of on Monday that binds to the A site on the 30S side of the ribosome? What other class did we make mention of? I did say aminoglycosides, good. Now, do aminoglycosides functionally shut down protein synthesis? Maybe, maybe not, but their major effect, their bactericidal effect, rests with their ability to cause misreading of that RNA template. Okay, so remember, aminoglycosides are going to bind to ribosomal RNA, and they're going to lead to misreading of the message. The ribosome is going to skip over codons or misread codons, leading to the production of these aberrant proteins that are going to go start poking holes and damaging the bacterial cell, leading to a cidal effect. Right, leading to a cidal effect. Good. What class binds to this 50S aspect? Maybe in the P site. We made mention of one class. There's several, but we focused in on one.
Macrolides.
Macrolides, very good. Macrolides, azithromycin, clarithromycin to a much lesser extent. I haven't dispensed erythromycin and I can't tell you how long, okay? But nonetheless, when a macrolide like azithromycin binds into this 50S P site, that interaction that wants to occur between the existing peptide in the P site and then the amino acid that just came in from the A site is inhibited. Remember, that charged transfer RNA has an amino acid attached to it. That amino acid, the amino group, the amino functionality is naked. It's nucleophilic. It wants to attack the peptide bond that exists or the ester bond that exists between the transfer RNA in the P site and that growing peptide strain. It wants to attack that, let that other transfer RNA go, and add itself to that growing strand. These drugs inhibit that process. They inhibit peptidyltransferase. Okay. There are others, clindamycin, if you will, a lincosamide, and then some streptogrammin derivatives that we may or may not make mention of throughout the course of the semester. But macrolides, and to a lesser extent, clindamycin are the ones that we need to focus in on. Okay. All right. Let's look up here real quick at the top. We have 2 pathways being shown here. Para-aminobenzoic acid getting converted to dihydrofolate, and then dihydrofolate getting converted to tetrahydrofolate. There were 2 enzymes that I made mention of. One of them is, what was it? Dihydrotoroate synthase, right? That particular enzyme is responsible for generating precursors to folic acid. And I made mention of one molecule that does a nice job of inhibiting that process. What was the name of that drug? Sulfamethoxazole. It is sulfamethoxazole. So the sulfa derivatives in general, and then specifically sulfamethoxazole, shut down that enzyme. They inhibit that enzyme, preventing then the production of the precursors to folic acid. Okay, if by chance that bacteria circumvents that process a little bit and is able to make some folic acid, it still has to reduce that folic acid down into tetrahydrofolate, and then subsequently the 1-methyl donor that leads to the formation of thymidine from uridine. The enzyme dihydrofolate reductase is a target of antibiotics, and we do have one particular molecule that is paired with sulfamethoxazole that you have all seen. What's the name of that drug that inhibits bacterial dihydrofolate reductase? Trimethoprim. Good. Trimethoprim. All right. So that gives us kind of a quick review of what we discussed. We're going to finish out then with this part of the slide, cell wall synthesis. And when we talk about cell wall synthesis, we have antibiotics that specifically inhibit the enzymes responsible for synthesizing cell walls, inhibit transpeptidation reactions. And then we have some other molecules that actually sequester starting materials of peptidoglycan synthesis. So we'll talk about both of those in a minute, okay? All right, what questions do you have though to this point? We doing okay? All right. Cell wall synthesis. We already made mention of the fact that bacteria and humans alike have a cell membrane, so it's very difficult for us to want to go poke holes in cell membranes because we lose a lot of specificity when it comes to a bacterial cell and then our own cells. However, since bacteria do have a cell wall, if they have a cell wall, they have to have enzymes that synthesize that cell wall. Since we don't have those enzymes, we can selectively target those bacterial enzymes, try and inhibit them, and then compromise the integrity of the cell wall, leading to bacterial cell death. Okay, and then if you like the pictures, again, too, this is just meant to give you some type of rendition about how thick a cell wall is in the gram-positive organism versus the gram-negative organism, okay? All right. What's the name of those enzymes, incidentally, that bacteria have that synthesize this cell wall? They're collectively referred to as penicillin binding proteins for obvious reasons. They bind to penicillins. And they come in a variety of different flavors. They have a variety of different molecular weights, and not all penicillin binding proteins participate in the same chemical reaction. Some of them are very, very important for expanding the bacterial cell wall. Some of them are responsible for forming a septum so that the bacteria can divide. Some of them, I call them molecular razors. You may have a bacterial cell with little bits of peptidoglycan sticking off the side, and one of the functions of a penicillin binding protein may be to kind of remove some of those whiskers, if you will, not the whole cell like I just did there, but you know, shave off a little bit here and a little bit there to get that smooth morphology, okay? When we inhibit penicillin binding proteins for therapeutic purposes, we want to inhibit penicillin binding protein 1, 2, and 3. Those tend to be some of the more critical penicillin binding proteins, in particular in gram-negative organisms. I'll tell you, I give up, you guys. There we go. All right. Tell me what peptidoglycan is actually made of. Sort of in the name. Peptido, meaning peptide, and then glycan, meaning sugar. So these are sugar polymers, N-acetylmuramic acid, glucosamine. They get polymerized together, okay? And then these sugar polymers are fortified and stitched together with amino acid fragments that come together as peptides. Okay? And then when you have them come together and zippered up under the direction of a penicillin binding protein, this is what gives the strength, the structure, and the rigidity to the bacterial cell. Why do the bacterial cells need a cell wall in the first place? What's the functional role of the cell wall in a gram-negative or gram-positive organism? Do they do this because it looks cool, or do they have a reason for doing it?
It's structured.
It is structure, right? Structure. What does structure lend to? Protection. Protection from what? Antibiotics. No, that's not a bad idea. Absolutely. But think about it a little bit more from a physiological perspective. In the absence of antibiotics, structure and protection, what is that bacterial cell wall protecting the cell from?
Other cells.
Other cells? Other cells, okay. What else?
It could be protecting it from the environment itself,
Okay,
from getting too hot and too cold.
all right. So again, too, when temperature increases and decreases, that can do weird things to the environment, to the cytosol, right? Expand, contract. Maybe that cell wall maintains some structure in that regard. What else? Yeah,
Provides rigidity.
rigidity. We're getting there. We're getting there. I just need someone to say it. Right? Rigidity, protection, protection. What's inside that cell? What's inside that bacterial cell? What is it chock full of? What's that?
DNA.
DNA, chromosome. What else? How would you characterize the inside of that cell from an osmolality perspective, osmolarity perspective, rather? Is it isoosmotic? Is it hyper, or is it hypoosmotic? Think about everything that that bacterial cell is concentrating inside itself, because all it really wants to do is replicate. So it's very, very hyperosmotic. And what do hyperosmotic cells want to do? Okay, what prevents them from lysing? Cell wall. If we compromise the cell wall somehow, that leaves that cell prone to lysis then, cell death. Okay, that's why it's really, really neat then that we can identify small molecules that inhibit this process. On the left side here in blue, we've got what I'm going to refer to as the C-terminal side of forming peptidoglycan. And in most bacteria, this is just comprised of two alanine residues, D-alanine, D-alanine. This is the C-terminus. On the right side, the pentaglycyl unit in gram-positive cells, all right? Adipic acid in gram-negative cells. So here's a pentaglycyl unit. And all of these gray boxes that you see up in here are meant to represent then these polymers of N-acetyl-glucosamine and then N-acetylmuramic acid, the sugar component. What the bacteria wants to do is it wants to link all of this up together to, again, fortify its cell wall. And essentially, the N-terminus from this pentaglycyl unit wants to bind with the carboxylic acid on the C-terminus to form an amide bond. If you took a carboxylic acid and you took a free amino group, threw them in a flask with some water and let them sit there, you would wait a long, very long time, and maybe you get an amide bond formed at some point. But this process gets sped up significantly under the catalysis of these penicillin binding proteins. Okay. And the penicillin binding protein, we'll look at this in more detail on the next slideshow, leads to the formation then of this amide bond, all right, that peptide bond. Peptide bonds are pretty stable. They're much more stable than an ester bond, okay? So that's the role of the penicillin binding proteins. We have three different classes of drugs that we'll refer to as beta lactams from here on out. This is a penicillin. That's a cephalosporin. And then that's a carbapenem. We're going to get into the structures of these in much more detail soon. But any one of those three drugs has the capacity then to inhibit a penicillin binding protein thereby then preventing the formation of that amide bond. Others to consider. Are there other mechanisms that we can use to inhibit cell wall synthesis? Yes, in fact, there are. Now, what's interesting about these latter two categories is that they don't necessarily target an enzyme. They don't target an enzyme, but rather what they're going to go after is the precursor molecules. Right? There's a class of drugs called glycopeptides. Vancomycin is the one that we use here in the United States. There's another one called teicoplanin that's used outside of the United States. It's a glycopeptide. And a glycopeptide like vancomycin has the ability to essentially cap D-alanine. It basically sequesters it. Okay, it sequesters it so that that C-terminal fragment isn't accessible to the enzyme itself for the purposes of making a peptide bond. Okay, so vancomycin's role is to cap the C-terminal end of D-alanine. Okay. Other drugs, and I think their use is going to start to increase. Lipoglycopeptides. They're called lipoglycopeptides for obvious reasons. They have a lipophilic anchor coming off their structure that helps to keep them in place in and around the bacterial cell target. One of them is, well, does anybody know the name of them? The lipoglycopeptides? Yeah. One more time. The vancins, yeah. Are you familiar with the vancin? Name of vancin. Dalbavancin, good. The vancins are fun to party with, I hear. Another one. Telavancin. VANCIN, good. There is one more that I think, I don't know how often it's used. Anybody know the third one? Vancin, good. All right, so these are the vancins. Bless you. And from a mechanistic perspective, they do the same thing. Okay, they do the same thing. They cap D-alanine, thereby preventing peptidoglycan synthesis. Okay. All right. We kind of went over this already, so I think you're in good shape there. Selective toxicity, everybody hopefully has some type of functional construct for them of what selective toxicity really refers to for the purposes of treating bacterial infections. But I want to also impress upon you that this concept is not just limited to bacterial infections. Later on in the semester, when we start talking about fungi, maybe even if you get into some viral infections and other infectious disease, the concept of selective toxicity is paramount to not only treating the patient, okay, getting a medical cure, but also decreasing adverse effects and toxicities. So it extends to antifungals, and parasitics, as the like. Okay. All right. So this is kind of like the complete list from that handout. We will elaborate on this molecule more so this semester than we had in the past, just because it's being seems to be being used a lot more. Okay. And that takes us to the end of our introductory material. I did have that handout from Monday. And at the bottom of that handout, it asked you guys to look for an investigational drug. Considering that you guys have a mini quiz on Monday, why don't we just put that off until next Wednesday then? Okay, if you have time between Monday and Wednesday morning to look up something and educate us on that. What questions do you have about what I've covered thus far then? All right. Awesome. I'm gonna get into some penicillins then. Okay. Objective wise, I will tell you, for all the antibiotics that I'm gonna start talking about, whether it's beta lactams or macrolides or whatnot, um, I'm a medicinal chemist. I pretend to be a pharmacologist and I'm a pharmacist. So what I want you guys to walk away with is not memorize the structures. Okay, I don't want you to spend a lot of time memorizing individual structures. Rather, what I want you to do is understand the pharmacophore of these various drugs. Right? When I say beta lactam, I automatically want you guys to think about a chemical system, a chemical structure. I want you to imagine what that beta lactam looks like in your mind. And then what's important is what we attach to that beta lactam to enhance its activity against gram-positive or gram-negative organisms, to protect itself against the hydrolytic effects of beta lactamases. Right? What has been done to that molecule to broaden its PK parameters? What has been done to that molecule that allows it to actually treat this otherwise resistant organism? So that's sort of the things that I want you to start cluing in on. If there is a specific moiety, a specific drug that I want you to commit to memory, I will tell you, all right? But it's the overarching concepts that I think are important. So mechanism of action and structure activity relationships, right? Those are the important things. Pharmacology wise, I'll make mention of some AD, ME, and T. And then this is where we're going to start really diving into the resistance, hopefully. Okay. So objective wise, it's going to be pretty similar whether we're talking about the penicillins, the cephalosporins, and the carbapenems and the like. Okay. So some of you may have seen this before. Some of you may have not. Culture growing from a patient suggests a penicillinase producing Staph aureus, not surprising. If you were to haphazardly administer drug molecule 1, drug molecule 2, or drug molecule 3, would any or all of those result in a treatment failure? Would any or all of them result in a treatment failure? Well, I don't really expect anybody to know the answer right now, but at the end of the session, hopefully you'll have a more informed approach to treating this patient. Okay? All right, my gosh, I'm having a deja vu moment. I think I just mentioned N-acetylglucosamine and N-acetylmuramic acid. And I think I just got done talking about how these polymers get cross-linked under the direction of penicillin binding proteins to form that peptidoglycan. We already talked about that and that. And then we did make mention of the rationale behind the cell wall in the first place. We're done with pictures for now. Let's review the cell wall synthesis approach very briefly, though. Cell wall synthesis in bacteria, it's a very, very complicated and prolonged sequence of events. There's over 30 steps, 30 enzymes and whatnot. And the majority of the synthesis of cell wall occurs in the cytoplasm, with the exception then of that final step. That transpeptidation step that occurs, which cross-links everything, zippers it together, does happen extracellularly. And we already made mention of the enzymes that are responsible for that, the penicillin binding proteins. Here's just another image. Again, too, historically, I apologize for the redundancy today. What I just finished up would have been done on Monday, and then we would have restarted this today. So that's why we're seeing a lot of repetition here. Nonetheless, here is the C terminus, and then here is the N terminus. And then these are brought together under the direction of these penicillin binding proteins. If we were to take a dive down into the active site of the penicillin binding proteins, what you would notice is that the active site has a serine residue at the center. That's probably the most important residue that exists in the active site. And it serves as the nucleophile that's going to initially attack that D-alanine, D-alanine C-terminal fragment of forming peptidoglycan, okay? After the serine residue attacks that D-alanine, D-alanine residue, what you end up with is a leaving group. An alanine residue gets broken off and leaves, and you formed this classical acyl enzyme intermediate, whereby the serine residue is attached to that penultimate, what was that penultimate? Alanine. And that interaction that exists between the serine and that alanine is an ester bond. That ester bond being exposed and being labile is just sitting there and waiting to get attacked by something. And what wants to attack it? The amino fragment, the amino group on that pentaglycyl unit on the opposite side of peptidoglycan. That amino group is going to come in, attack the carbonyl carbon in the ester function, hydrolyze the ester, release the serine, and now we have formed an amide bond. Here's the serine residue in the active site. Not shown here, but there is a basic residue, a histidine residue that's going to pull off a proton from the hydroxyl group, generating this very, very nucleophilic alkoxide anion, which is going to go up and attack the carbonyl carbon in the amide bond. And this alanine residue is going to leave our leaving group. And here's our acyl enzyme intermediate. Okay, here's the serine residue, and it's formed this ester bond with the C-terminal side of peptidoglycan. And then on the opposite side over here, which isn't shown in the top right, but is shown here, is that pentaglycyl unit. The amino group is nucleophilic. It's got a lone pair of electrons. It's going to want to come in and attack the carbon, the carbonyl carbon in that ester. That'll ultimately lead to ester hydrolysis. The nitrogen has attacked the carbonyl carbon, formed an amide bond, and then literally regenerated the serine active site. Okay, and this is what happens then over and over and over in the active site of various penicillin binding proteins that are responsible for that transpeptidation reaction. Okay, so why not inhibit those? What happens if we inhibit those? If this is a cocci, a coccus, right? And here's the cell wall. Under the influence of beta-lactams, what you end up with then, a cell wall that starts to look like this. And then it starts to look like this. And then the cell wall has completely disintegrated, and you're left with this spheroid, which is basically a naked bacterial cell. No cell wall, just cell membrane. And then what happens is it explodes under the pressure, okay? So that's what we want to happen. I already told you that these enzymes come in different flavors, different molecular sizes, and high molecular weights seem to be responsible for some more of the more important chemical reactions with regard to cell wall synthesis. I think we've hit on this, so I don't need to spend time on that slide. That takes us to penicillins then. Penicillin discovery. Pretty serendipitous if you ask me. So it was discovered through some sloppy scientific work. And penicillin was discovered to be synthesized in molds under the direction of an enzyme called ACV synthase. So ACV synthase is an enzyme that will take a couple amino acids, cysteine, valine, and then something called adipic acid. It will put these 3 amino acids together in a cauldron, stir it up a little bit, say a spell, and poof, what you end up with is something that looks like penicillin N. Penicillin N was one of the first penicillin derivatives that was discovered. Penicillium nodosum is where the N comes from, I believe. And when you look at the structure of that molecule on the bottom right, you see the classical penicillin pharmacophore And then attached to the pharmacophore is this long, wavy, flexible side chain coming off what will soon be discovered as the sixth position. So this molecule is regarded as weakly active. So penicillium, it's a mold. Right? These molds are making these metabolites. These metabolites can be isolated and purified and then packaged up and then given to patients who have infection. Right? So this is done in a fermentation chamber, right? A vat. You grow all of this mold, feed it the right starting materials, it spits out the penicillin. Okay? So as such, the classical older penicillins are referred to as fermentation-derived penicillins. Do we want to make a lot of this? No, we don't, because this is weakly active. But we know that if the mold is using something called adipic acid to add on to the backside of the penicillin pharmacophore, what if we replace that adipic acid with a different type of carboxylic acid? What if we supply the mold with some other starting material? Can we make derivatives? And that's in fact what happened then with drugs like penicillin V and then the injectable penicillin G that you may or may not have seen at your practice site. You swap out the adipic acid for phenylacetic acid or something like phenoxyacetic acid. You put that different acid substrate into the vat, and then it will generate for you the desired penicillin. All right, so here is phenylacetic acid. Throw it in with your mold. And what do you end up with? Pen-G, penicillin G, or phenoxyacetic acid. Throw that into the culture medium. Let the mold use that to synthesize your penicillin derivative. And then you end up with the orally used penicillin V. And so these are fermentation-derived penicillins. You kind of run out of carboxylic acid substrates to throw into the mold culture, so you have to become a little bit more creative when you're trying to discover derivatives. And one approach that was taken to align then with this semi-synthetic direction to making drugs was taking that penicillin N, let the mold make up a lot of penicillin N, isolate the penicillin N, and then treat the penicillin N, excuse me, with an enzyme that will remove that weak side chain. And what we end up with, you guys, is a new and unique starting material called 6-amino penicillinic acid. 6-amino penicillinic acid. Still maintain this very important pharmacophore that we'll talk about in a minute. Now on the back end, here at the sixth position, you have an amino group. To that amino group, we can attach all sorts of side chains to impart a lot of diversity then on your penicillin derivatives. And when we do that, that's our semi-synthetic approach to making penicillins. Penicillins historically have been used to treat a variety of infections, gram-positive infections, gram-negative infections as well. However, I think resistance mechanisms, in particular in the gram-negative realm, has limited their use. There are some gram-positive organisms that yet today still remain exquisitely sensitive to penicillins. Okay. There is no shortage of penicillins that have been discovered and used clinically over the years, but not all of them are used. So despite the preponderance of drug, the sheer number, we're going to focus in on the ones that I think are clinically relevant. And Dr. Lewis will add to that or subtract from that based on her experience as well. Okay, but what's important though is you really need to know when you can use a penicillin and when you shouldn't use a penicillin. Up front, sick patient, not really sure what's going on, kind of have to be a little bit broad with your empiric therapy, but when those results come back, and it says it's sensitive to penicillin, good amount of penicillin. Okay. The fermentation derivatives I already made mention of, Pen V and then Pen G. There is a subset of penicillins that we call penicillin ACE resistant penicillins. Nafcillin and then oxacillin are two representatives of that class. We refer to these guys as anti-staph penicillins, and I'll tell you why in a little bit. And then we have semi-synthetic penicillinase sensitive penicillins that are somewhat broad in spectrum. Piperacillin is a good example of that. And then there's two aminopenicillins, ampicillin and then amoxicillin, that we'll make mention of as well. Right, but mechanistically, what are these drugs really doing? Oh man, I couldn't hear my alarm. Let's take a break, you guys. I'm sorry. I had an alarm set for 9:50. I didn't hear it. Let's take a break, 5 to 7 minutes or so, and then we'll come back and pick it up, okay? Right. Hey, how are you? Good. Good. What's going on? Good.
Antibacterials.
Yeah.
Beautiful.
Yeah, no doubt.
Yeah. Good old days, right? That's what I remember.
Maybe.
When they're
Maybe.
designing those. Yeah, I mentioned these briefly in case, you know.
Let's go ahead and pick it up then. So we're talking about beta-lactams and specifically penicillins. What I will impress upon you though is regardless of what beta-lactam we're talking about, whether or not it's a penicillin, a cephalosporin, or a carbapenem, mechanistically, it's the same, okay? Beta-lactam antibiotics are irreversible inhibitors of penicillin binding proteins, and they specifically want to covalently modify that serine residue that I showed you earlier on, okay? This leads to that bactericidal effect, okay? As the cell wall breaks down, secondary to the inhibition of the penicillin binding proteins, and then that spheroid forms and sort of pushes itself out. The hypertonic inside the cytosol of the bacterial cell causes it to lyse. Right? And how do they accomplish this? What makes beta-lactam get accepted into the active site of the penicillin binding protein in the first place? Well, it's because these drugs have some resemblance to that C-terminal fragment. They look like D-alanine dealanine. So they're essentially tricking the enzyme into accepting it into the active site, whereby the serine is going to act upon it, okay? And then cumulatively, that prevents transpeptidation reactions. So here is a rendition then of a penicillin binding protein active site. And what I'd like to show you here is, well, before we do this, hello. Okay, who's getting dizzy? There we go. So when we say beta-lactam, first of all, what is a lactam? I know some of the folks in the audience have been asked this question before, but what is a lactam? What is a lactone? So when you hear beta-lactam, everybody uses the word beta-lactam out in the community, but not everybody understands what they're really saying. When you hear beta-lactam, what I really want you to think about is this four-membered ring. Right? It's a cyclic amide. It's a high bond, it's been cyclized. Right? That's a beta-lactam. If you extend it by one carbon, right, you go from beta to gamma. You extend it by one carbon again, then you have a delta-lactam and so on. Right? So these are cyclic amides. And then a lactone is a cyclic ester. Okay, a cyclic ester. The macrolides are cyclic esters. You see beta-lactam or hear beta-lactam, that's what we should be having on our mind. And remember, these penicillin derivatives are de-alanine mimetics. What is the side chain in alanine? Think back to your biochemistry. What's the R group in an alanine residue? So it's a methyl group. Okay. It's a methyl group. The penicillin of your choice binds into the active site. When that penicillin binds, that leads to then the initiation of an acid-base catalyzed reaction. Glutamic acid residue is going to serve as a general base. It pulls off a proton from the histidine. The histidine is going to pull off the base from the serine. The serine residue is going to come up and attack the carbonyl carbon, right, in the beta-lactam bond. It's going to break that beta-lactam bond, and in doing so, what it does is it covalently modifies the enzyme active site. And so look here. We went from that intact beta-lactam up to the top to this opened ring where the serine residue is now been esterified with the drug. Is this a covalent interaction? Is this covalent? This is a covalent bond. It's an ester. Esters are covalent. Amides are stronger, but nonetheless, this is still covalent. If this is here, transpeptidation reactions can occur. Let me ask you another question then. Thinking back to the normal approach to peptidoglycan synthesis, what hydrolyzes that acyl enzyme intermediate during transpeptidation reactions in the absence of a drug? Remember, the C-terminus just acylated the enzyme active site and an alanine residue was lost. What's the next step that leads to transpeptidation in the absence of drug?
The N-terminus of the other side of the peptidoglycan.
Absolutely, good, good. And so that pentaglycyl unit comes in and attacks the ester bond, hydrolyzes the ester, leading to the formation of the amide bond. Why doesn't that happen here? This five-membered heterocyclic ring, this thiazolidine ring in the penicillins, along with these two methyl groups, produces a significant amount of steric bulk in the active site. And because of that bulk, that pentaglycyl unit can't come in and get access to this ester bond to hydrolyze it off to regenerate the penicillin binding protein active site. Okay. So the bulk here is important because it prevents then hydrolysis, right, of this inhibited enzyme active site. This is an old and cruddy looking picture, but I kind of like it because you can line up a penicillin and you can line up a D-alanine residue kind of nicely from an electronic perspective. Again, the drugs themselves are D-alanine mimetics. So if this is a D-alanine mimetic at the bottom, here's a carboxylic acid. And here's a carboxylic acid in the penicillin derivative. Those are actually important for establishing then an ionic bridge with a lysine residue in the active site. Here is the critical beta-lactam bond, or excuse me, here's the critical amide bond that gets broken during normal peptidoglycan synthesis. And then here's the beta-lactam bond that gets opened up when the serine residue in the active site attacks a penicillin. Here's a methyl group. Here's a methyl group. Here's another methyl group. Somewhere in here, there's another methyl group. So the methyl groups sort of align spatially to trick that enzyme into accepting the penicillin into the active site. Okay, so there's just some good structural agreement. You could almost take the two and superimpose them upon each other and see some nice agreement from a lipophilic perspective and then an electronic perspective. So what's the cumulative effect here? A stable ester bond is formed. There is no leaving group that is generated during this particular process that induces the bulk that prevents the hydrolysis of the acyl enzyme intermediate. I thank the sterics for that. Let's talk about some structure activity really quick. I don't know why my symbols don't render correctly on my slideshow. I apologize for that. Let's go through here and just highlight a couple important aspects of the penicillin. All right, so there's the beta-lactam unit, and this particular bond right here is by far the most critical bond, most critical aspect of the drug. If that hydrolyzes, you will not have any activity. So if that goes, you're done, okay? So that's important. The carboxylic acid, again, mimics the C-terminus and D-alanine D-alanine, and it establishes an ionic interaction with a residue in the enzyme active site. The methyl groups, again, too, they're there because they help to make it look like D-alanyl D-alanine. There are two protons at the 5 and 6 position of the beta-lactam unit. They're in the alpha configuration, so they're pointing away from you. Those are required for activity. That stereochemistry does become important, but those alpha protons take up a lot of room on the slides. So from here on out, I'm going to omit them. Just assume that they're there, and I probably won't ask you about them ever, okay? And then coming off the 6th position, we have this amino group. To that amino group, this is where all the magic occurs. We can attach so much diversity to the amino group off the 6th position that our group could represent just about anything your imagination can take you to. Okay? And a lot of our groups, a lot of diversity is tolerated with regard to the beta-lactams, the penicillins. Some of the R groups that we'll see help to enhance resistance to penicillinases. Some of the R groups that we see help to enhance the spectrum of activity. You know, do you want a gram-positive coverage? Do you want gram-negative coverage? Do you want both? The R groups a lot of times can dictate which is which, okay? And then that takes us to resistance. In a broad sense, what is a beta-lactamase? How would you just, yep, go ahead.
Sorry, it's something that breaks the beta-lactam bond.
Good, good. So it's an enzyme. All right, so it's an enzyme, first and foremost. Okay. This is an enzyme that has the capacity to chop that critical beta-lactam bond, rendering the drug inactive. okay? When we say beta lactamases, that, my friends, is an umbrella term. Okay? Beta lactamases, that's up here. Now, under the umbrella exists so many different types of enzymes. It's mind-boggling to try and keep straight, okay? We have beta lactamases that are penicillinases, cephalosporinases, and then carbapenemases, okay? And the substrate specificity is implied by the name, okay? But it's very, very tricky. It's very, very tricky because some carbapenemases, right, have the ability to hydrolyze any and all beta lactamases. And conversely, some penicillinases will only hydrolyze penicillins, but not cephalosporins or carbapenems. Okay, so we have to start thinking logically about how we're going to address this problem with beta lactamases. And my approach to discussing beta lactamases is to subscribe to the Ambler classification. You guys just put your pens down and just listen for a couple minutes. Let's just talk about this, and then I'll summarize what I want you to know from here, because the direction that ID is going, this is going to be more and more critical to understand when you guys get out there and start practicing as compared to when I was in the hospital. Most of what I'm going to talk about today was still out in the realm. Nobody really understood what was going on. Okay. The Ambler classification, though, itself has been around for a long time. And this is a scheme that looks at the amino acid sequence of various beta lactamases. Right? It looks at the primary sequence. And then it lumps these enzymes together based on the homology that exists amongst these different enzymes. Okay. And the Ambler classification divides enzymes up into 4 flavors: A, B, C, and D. Okay, so we have class A enzymes, which are penicillinases, but also within this class, you find the extended spectrum beta lactamases. You find things like KPC, which is a carbapenemase. Right? So there's a couple examples right there. And then this is a serine beta lactamase. I'll come back to that in a moment. The class B enzymes are represented by something called the New Delhi metallo beta lactamase. There's an enzyme called VIM, V-I-M. There's an enzyme called IMP, right? And in the active site of the class B enzymes, we don't have a serine residue that's serving as the nucleophile, but instead a water molecule is the nucleophile, all right? So we call these metallo beta lactamases. These are particularly concerning because you guys know who the honey badger is? You're familiar with the honey badger? Yeah, the honey badger just doesn't care. The class B enzymes do not care. They don't care if it's a penicillin, a cephalosporin, or a carbapenem. They will chew it up and spit it out, all right, and hydrolyze it. They're concerning because they're starting to become more and more common in areas where you never really saw them, okay? Dr. Lewis will tell us horror stories about what she's encountered at her practice site with regard to some of these resistance enzymes. I can't wait to hear. We have class C enzymes. The class C enzymes are kind of easy because the C does not stand for cookie, it stands for cephalosporin. You guys don't like my jokes. I need some new jokes, don't I? All right, so these are cephalosporinases. And AMP-C is probably one that you've already heard of, okay? And then the class D enzymes, these are the oxa-like oxa-like beta lactamases. So OXA 10, OXA 4, OXA 1, these were the first recognized enzymes that had the ability to chew up oxacillin. But now what we're finding out is that some of these more recent discoveries, unfortunately, identify enzymes that can chew up carbapenems as well. Okay, so OXA 48, for example, is a very concerning class D beta lactamase because it can hydrolyze certain carbapenems. So 3 of the 4 are serine-based enzymes. And that's important to know because we can inhibit some, if not all of those, with various beta lactamase inhibitors. Okay. All right. So just kind of, take a snapshot of this in your mind. And we're going to keep coming, bless you, keep coming back to this. Okay? Because at some point in time, you're going to get presented with a scenario where a patient has a difficult to treat infection, and you come to realize that they're expressing that enzyme, the organism that is expressing that enzyme and this enzyme or that enzyme. How will you go about treating that patient? Or what if it's got an ESBL and then a metallo beta lactamase co-expressing? This creates a lot of anxiety for people in the infectious disease world, right? But nonetheless, what is the role, what is the approach then to these beta lactamases? Why did bacteria evolve them in the first place? Well, again, too, beta lactamase enzyme was thought to be a mutant penicillin binding protein since they look very similar in structure and composition to the penicillin binding proteins. And when you look at what's going on inside the active site of the class A, C, and D enzymes, you will find a serine residue in the active site, and that serine residue does get activated much like the penicillin binding proteins, ultimately generating that alkoxide anion, which then goes in, attacks the carbonyl carbon in the beta lactam bond, opens it up, and then what's not shown here is what we refer to as the deacylating water molecule. After the serine residue has attacked the beta lactam and formed that stable ester bond, there is no pentaglycyl unit in the lactamase enzyme coming over to release it. Rather, what these enzymes have is a deacylating water equivalent that comes in and attacks the ester bond, hydrolyzes it, and then regenerates the active site. Okay, so mechanistically very similar with a little bit of nuance here at the end with the deacylating water. And what I want to show you here, since we are talking about penicillins, so the penicillinases, the enzymes that are classically described as penicillinases, typically have a very, very narrow entry point to their active site. Okay, so a small molecule like a naturally occurring or fermentation-derived penicillin can slip into that active site relatively easily and then get hydrolyzed. Okay, so sort of a defining feature of some of the earlier classical penicillinases, much like the BLAZ product in Staph aureus, the PC1 beta lactamase.
So is it that narrow active site that gives it a little more specificity versus inhibiting its own cell wall synthesis?
So I got the first part of your statement, but then the inhibiting the cell wall synthesis part, help me out with that.
If it is a mutant of the PBP, and would it be potentially able to?
I got you. Okay, I understand. So 2 points about that. One, I hadn't thought about that, but yes, I don't necessarily disagree with that. When you think about the real estate that is required to catalyze transpeptidation reactions, you need a pretty much a larger open space, okay? Number two, what's interesting about the beta lactamases that bacteria synthesize is that they secrete them. A gram-positive organism that encodes a beta lactamase is going to export that into the immediate extracellular environment, okay? So they're not membrane bound. The penicillin binding proteins that catalyze transpeptidation reactions are membrane bound proteins, okay? In a gram-negative organism, what's that space? What was that space called that exists between the cell wall and then the outer membrane? The periplasmic space. What do you think is inside that periplasmic space floating around? Beta lactamases. Yeah, okay. So they are mutant, okay? They are not membrane bound. They're free floating, if you will, right? They're sequestered in the periplasmic space for obvious reasons. If the penicillin binding protein is localized in that membrane, okay, let's have enzymes that can degrade drug that might inhibit it at the site of action. Gram-positive organism, let's secrete beta lactamases in the immediate environment to degrade anything that might come in and harm us or want to take our space or our resources and kill us, okay? But I agree on both accounts. Yep. The narrowness of the active site and the entry to the active site dictates or predicts only a certain amount of drug is going to make its way in there. Why is that important then? If a penicillinase, all right, can degrade a drug like Pen-G or Pen-V, and you know that that organism is synthesizing and secreting that, what does that automatically tell you you're going to do? You're not going to use Pen-G or Pen-V. You're going to find something else that might have a larger profile, either on the back end or the front end. And we'll talk about that in a second. Okay. Does this make sense to everybody? Are we good with this? Okay, are we good with this concept of beta lactamases then? There we go. And then when we talk about the class B enzymes, so the class B enzymes do not have a serine residue in their active site. Rather, they utilize a zinc coordinated water molecule to serve as the nucleophile, okay? So by way of example, here is the active site of New Delhi metallobeta lactamase. And inside that active site, you'll see some zinc ions coordinated in the active site. And those zinc ions then are positioning and activating an hydroxide anion. When the carbapenem or the cephalosporin or penicillin binds into the active site of this metallobeta lactamase, the water molecule is going to come up and attack. You have a transition state formed. The electrons are going to flow back down into the carbonyl carbon. And then the beta lactam bond that was formed by sharing those two electrons those electrons are going to fall back on the nitrogen. You end up with then this hydrolysis product still bound in the active site by the coordination of these zinc ions. And then water, of all things again, is going to come in and hydrolyze the ester bond that's coordinated between the zinc and the molecule and regenerate the active site. Okay, and that happens over and over and over. Okay, so the main difference then between the serine beta lactamases and the metallobeta lactamases is the nucleophile. Okay. And at some point in time, if we get to it, what we'll do is we'll talk about how we are addressing inhibiting metallobeta lactamases these days with chelating agents of all things. We've got some interesting inhibitors out there that have the capacity to chelate the zinc and then prevent it from coordinating water and thereby inhibiting it. So mechanistically, acylate the enzyme's active site, shut down transpeptidation. Resistance mechanisms, the most clinically relevant form of resistance is hydrolysis by beta-lactamases. Okay. The fermentation-derived derivatives, PEN-G, PEN-V, again, they're either oral, PEN-G, PEN-V, or IM or IV for PEN-G. The problem with these drugs, in particular PEN-G, is that it's very sensitive to acid degradation in the stomach. Okay, so acid will hasten its destruction, and as such, PEN-G is given IM or IV only. PEN-G, the extra oxygen in the side chain actually imparts a little bit more of an electron withdrawing effect on the side chain that pulls electron density a little bit away from the carbonyl group in the side chain. That creates a little bit more acid stability to PEN-G relative to PEN-V, to PEN-V relative to PEN-G. So you can give PEN-V orally. And either way, regardless of which one you're talking about, PEN-V or PEN-G, these are very nice substrates for penicillinases, right? Penicillinases. So PEN-V, about 60% after oral administration is going to get absorbed. And penicillins do distribute pretty well throughout the body. They don't necessarily undergo any specific metabolism. They will get hydrolyzed to penicilloic acid, which is an inactive metabolite. And then they get excreted through the urinary tract, tubular secretion in particular. I already mentioned that. And then again, too, this sort of takes us back then to our initial slide in question, Staph aureus. The majority of Staph aureus today have a gene called the BLA-Z gene, beta-lactamase Z gene, which encodes a protein called PC1. PC1 is a penicillinase, right? And this penicillinase does a really nice job of hydrolyzing PEN-G, PEN-V, and a variety of other penicillins. So that's the principal mechanism of resistance for gram-positive organisms. Gram-negative organisms do also synthesize and secrete beta-lactamases into the periplasmic space, but efflux pumps are widely expressed in a number of resistant gram-negative organisms. They know that that penicillin or that beta-lactam is in the periplasmic space, wants to grab it from the periplasmic space and throw it back out into the environment. If it's not in the periplasmic space, it cannot inhibit the penicillin binding protein. And then porin modulation as well. Remember, what organism has porins, gram-positive or gram-negative? The gram-negatives. And the porins are these channels that are expressed in the outer membrane that allow the transport and passage of substances, water-soluble substances. If your gram-negative organism alters the structure of the porin or decreases the expression of the porin, the beta-lactam isn't going to gain entry into the periplasmic space. That can also culminate in resistance. I think this is kind of a great chance to talk about one other aspect really quick. When we have beta-lactams, and the lab goes and tests a beta-lactam against a gram-negative organism, what that lab can report is whether or not it's susceptible, intermediate, or resistant. Okay, and it might also give you some MIC data. But if it comes back resistant, a lot of labs aren't going to be able to tell you why it's resistant. It just reports resistance. Is the bug resistant to the drug because it's expressing an enzyme? Right? Is the bug resistant to the drug because it has efflux pumps? Or is the bug resistant to the drug because it modulated or suppressed the expression of porins? So sometimes you don't really know.
Nowadays, there's all of this sophisticated molecular technology that allows the lab to analyze the bacteria and literally detect the absence or presence of certain genes. The labs, I don't know, you guys have a BioFire? Is that one? So, are you guys familiar with the BioFire PCR multiplex instrument? What they can do is they can grow the bacteria and then basically assay the bacteria for the presence of a number of different genes. And you could detect the presence of AMP-C or the SBL or maybe a carbapenemase. Okay. But what if the assay says it doesn't have this, that, or that, and it's still resistant? What does that really suggest?
Genotype, it's wild type, doesn't have any resistance mechanisms. Phenotypically, it's resistant to your drug. How do you account for that? Well, chances are it might be because of porn modification. What is the name of the beta-lactamase that Staph aureus produces? What's that? The G?
AG. The back of AG.
You were saying PC1, right? That's what I thought I heard. PC1. Staph aureus, 90+% of Staph aureus isolated from the environment have the BLAZ gene, which produces PC1. PC1 predicts resistance to penicillins, most penicillins. Okay. We're not going to use Pen-G or Pen-V up front to treat a staph infection unless you absolutely know that staph is sensitive to fermentation-derived penicillins. Probably not going to happen. Okay. All right. What about strep? Streptococci. Strep gram positive or gram negative? Chains or pairs? Clusters, Coccobacillus, not all at once. Positive or negative? What was the question?
Gram positive or gram negative? Strep.
Gram positive.
Good, gram positive. How would you expect them to appear on a slide? Purple. Chains, pairs, clusters, pairs. Good. All right. So you have gram-positive cocci in chains. And you start to see increasing MICs to penicillin in streptococci. What's going on in streptococci that's leading to these elevated doses of, say, amoxicillin that are required to treat something like a group A strep infection? What I'm trying to do is get you to think about how do strep acquire, develop, or have resistance to penicillins? Excuse me. What's the mechanism of resistance to penicillins and streptococci? What could it be? Could it be efflux pumps? Could be. How many of you think efflux pumps is a reasonable suggestion for resistance to strep, penicillin strep? Why not?
Because efflux pumps are for gram-negative cells that are membranes, since you don't have an outer membrane direct access to them. Okay, good. Yeah. The target penicillin binding protein is actually on the outer surface of the cell. The penicillin doesn't need to get inside the cell. If it did get inside the cell and it got pumped out, that bacteria would be doing us a favor, wouldn't it? Putting it right where we want it to be. So let's eliminate efflux pumps as a resistance mechanism for the beta-lactams in gram-positive organisms. Okay. Now let's shift our attention to other approaches. What's another mechanism of resistance that strep might have to get around the presence of penicillin? This group over here that I've been neglecting all semester long. Any thoughts? Penicillinates. That's always going to be a great idea. Is the organism expressing and secreting a penicillinase, a beta-lactamase into the environment to degrade the penicillin before it even has a chance to bind and inhibit? You know what's interesting about strep? Don't code for beta-lactamase. You get that augmentin prescription for group A strep. Unnecessary. Okay. So if I tell you they don't make a beta-lactamase, and efflux isn't really relevant here, what else could be going on?
The genes that penicillin binding proteins.
Perfect. I love it. Mutations.
Mutations in the penicillin binding protein. These mutations that have occurred in strep over the years has decreased the affinity of the penicillins to their target in strep. That's why we have had to start giving higher and higher doses. What is it, like 90 mg per kg per day or something, Dr. Lewis? I don't know. Hi, how are you? Sorry. Nonetheless, mutations is the correct answer. Do you drink coffee? We'll go have coffee sometime. Shown here, penicillin binding protein in a wild type strain of strep. Penicillin binding protein 1A, 1B, 2A, 2B, and then 3. Okay. Look at this particular lane on this Western analysis, which looks at proteins. Okay. This particular resistant strain of strep is no longer expressing 1A and hardly is expressing 1B. It's suppressing the expression of 2A and 2B, but it is expressing 3. Okay, so it is changing the quantity and quality of its penicillin binding protein profile, still surviving, but not dying necessarily in the face of beta-lactams like penicillin. Okay, so I want you to remember this because this is kind of important. Okay, streptococci don't make a beta-lactamase. If you know it's a strep infection, you don't need to have a beta-lactam, beta-lactamase inhibitor for treatment, okay? Strep, group A strep in particular, still exquisitely sensitive to penicillins. Sometimes you need to just jack up the dose a little bit. Questions about that? Make sense? All of you? Okay. And then that takes us to these semi-synthetic derivatives. So what we've talked about today are some fermentation-derived derivatives. Now we have semi-synthetic penicillin derivatives, nafcillin, oxacillin, dicloxacillin. These are the 3 drugs that are probably still somewhat clinically relevant today with regard to contemporary treatment. Nafcillin and oxacillin are given parenterally. We still do use these, in particular when we have known Staph aureus infections. What you're going to notice about these particular 2 derivatives is the amount of steric bulk that exists. I didn't mean to do that. The amount of steric bulk that exists on the back end of the molecule, okay? In fact, let's just look at it really quick. Nafcillin or Nafcil is a parenteral anti-staphylococcal penicillin. It's shown here on the right. And when you look at the ring system, you hopefully are starting to recognize that as the pharmacophore of the penicillins. Right? It's got the beta-lactam bond. It's got this thiazolidine ring that we made mention of earlier. It's got these 2 methyl groups and then this carboxylic acid, which is important for binding as well. Right? That's the pharmacophore. What's coming off the back end, though, of this molecule is an ethoxynaphthalene ring system. It's all this bulk right here. This is a pretty large and offensive looking side chain. And this large offensive looking side chain has purpose, okay? This large offensive side chain does 2 things. Number 1, based on size alone, this may prevent the penicillin derivative from gaining access and slipping into the penicillinase active site that Staph aureus makes. Okay, so we have some size exclusion perhaps going on here with regard to this large back end side chain. The other thing that has been suggested is that this ring system actually protects attack of the serine residue on the beta-lactam carbonyl carbon. The serine residue in the enzyme active site of the beta-lactamase, it certainly wants to try and get here so it can hydrolyze that bond. But what we see is that the ring system, which does not have free rotation, is blocking that. So with nafcillin, this semi-synthetic penicillin derivative, that we also call an anti-staphylococcal penicillin, right? This semi-synthetic anti-staphylococcal penicillin is resistant to the hydrolytic effects of penicillinases. Okay. Its early predecessor and the prototype in this particular anti-staphylococcal class of penicillins was referred to as methicillin. And methicillin, much like nafcillin, has got a significant amount of bulk on its back end, doing pretty much the same thing, preventing it from gaining access to the active site, preventing it from aligning in the active site correctly, and protecting then the beta-lactam bond from attack of the serine equivalent in the active site. Okay. So methicillin led to the inspiration for nafcillin and oxacillin and dicloxacillin. But methicillin is antiquated nowadays. It's got some toxicities associated with it, which precludes its use in contemporary medicine. Okay. All right. So what's the important aspect of nafcillin with regard to treating staph infections? You had an exam question that said, name 2 reasons why nafcillin might be beneficial in treating a staph infection. Number 1, size. Right? Size matters here. Number 2, stability. If size precludes its entry into the active site, if it forces its way in there, then the stability imparted by that shield, that nafcillin ring system, protects it. Okay? And you can extend that concept then to oxacillin and dicloxacillin as well. Dicloxacillin is an oral agent. Dr. Lewis, have you used dicloxacillin anytime recent? Do you even see that anymore as an oral option for treating staph infections?
Once in a while.
Once in a while? Have any of you guys ever seen dicloxacillin at your site? You have, Olivia?
Yeah, I just dispensed it like last week.
Get out of here.
Yeah, I'm serious.
Seriously?
I don't know what we were treating, but they called it over and we were, I was like, we can have this. And it was like, we did. It was like hidden on the shelf, dust everywhere.
I love it. That's what I was gonna ask. Was it in date? So that's good. And you know what? I guarantee you, they were using it to treat a staph infection, because that's pretty much the only reason why you would use it. And it might have been like ecthyma or impetigo. We'll come back to that too. Very exciting. I'm getting excited for skin and soft tissue infections. This is gonna be great. It's gonna be a great semester. All right. Nafcillin. Man, we're running out of time. I got a couple minutes left. Would you allow me to talk about MRSA for a couple minutes before we part ways today? Do you mind? All right. MRSA. What is MRSA? It's an acronym. What does it stand for? Methicillin resistant staph aureus. Okay. All right. When we say methicillin, we saw what methicillin looked like a couple minutes ago. Methicillin is an antiquated anti-staphylococcal penicillin. Okay. Now, staph aureus, gram-positive cocci, typically gets around the presence of most beta-lactams by synthesizing and secreting a beta-lactamase enzyme to chop up beta-lactams like various penicillins. However, there is a subset of staph out there. It's not really even a subset anymore. It might be, the predominant species out there growing. Staph aureus today in the community and in the hospital have acquired a gene called the MECA gene. There's this genetic element, there's this cassette, if you will, that gets into the chromosome of staph. And one of the genes in this cassette is the MECA gene. And the MECA gene is responsible for encoding a protein, an enzyme called penicillin binding protein 2A. Okay. MRSA, methicillin-resistant Staph aureus, has a gene called the MECA gene. The MECA gene encodes for a transpeptidase enzyme called penicillin binding protein 2A. Who cares? All of you do. Okay. And you care because this particular transpeptidase enzyme, this particular penicillin binding protein is capable of synthesizing peptidoglycan in the presence of almost all beta lactams. Okay. Thank you. It's a catchy tune. Okay. I want you to think about this as we get into next week, Monday, all right? MRSA means MECA positive. MECA positive means the presence of penicillin binding protein 2A. Penicillin binding protein 2A means that Staph aureus isotype is going to be able to grow, divide, and proliferate in the presence of just about every single beta lactam that we have. Okay, they are insensitive to beta lactams. They're penicillin binding protein 1, 2, 3, 4 sensitive, but they make 2A as well, and that gets around the inhibition of the others. All right, I'm going to show you next week then what happens with this particular enzyme, and then what we've discovered get around its presence with at least 1 or 2 cephalosporins. Okay. All right. Any announcements or anything left, Dr. Lewis? We good?
If you have any questions, reach out to me. And do some Zoom session